Hello everyone, my name is Haneesh Jasuja and the title of my poster is Progression of Bone Metastatic Prostate Cancer Assessed by a 3D In Vitro Dynamic Model. So the prostate cancer is the second most common cancer in men in United States, and the key statistics shows that 1 in 9 men will be diagnosed with prostate cancer in their lifetime and 1 in 41 men will die of prostate cancer. And there are four stages of prostate cancer, and it is commonly seen that the high mortality rate is due to advanced stage prostate cancer, where cancer cells metastasize to the bone and interfere with bone remodeling process. And when these prostate cancer cells establish themselves at the bone site, they started experience continuous mechanical force by the interstitial fluid flow, and that is in terms of fluid induced shear stress. So, in order to understand the effect of interstitial fluid flow on bone development and the growth of bone metastasized prostate cancer cells, we created a 3D dynamic in vitro model. So, here we designed and fabricated a perfusion bioreactor that facilitates continuous fluid flow condition to the cells at a velocity corresponds to interstitial fluid velocity. We also utilize 3D nanoclay based bone mimetic scaffolds for bone generation from mesenchymal stem cells and to recapitulate prostate cancer metastasis to bone. So here we compare the results of 3D dynamic samples and to the static samples. And if we look at the results, we observe that mesenchymal stem cells grow faster under perfusion bioreactor than static culture. Moreover, we observed high differentiation rate of mesenchymal stem cells to bone cells under dynamic conditions that we assessed by osteogenic biomarkers such as RUNX2, ALP, and OCN. And lastly, we observed a directional orientation of mesenchymal stem cells under dynamic condition compared to static culture. So after seeding prostate cancer cells onto bone, we observed that there is an increase in E-cadherin expression in prostate cancer cells over time under dynamic conditions. That helps them to bind tightly to each other. We also observed that MDA-PCA2B cells make compact tumors under continuous fluid flow compared to static culture, where we observed grape-like structures of the cancer cells. So from this, we can clearly say that there is an impact of continuous fluid flow on bone development and cancer cell morphology and their gene expressions. So overall, we can conclude that interstitial fluid flow has a critical impact on bone metastatic prostate cancer progression, and the perfusion bioreactor and nanoclay-based in vitro model could recapitulate the prostate cancer metastasis to bone under continuous fluid flow conditions. Thanks.